We are talking at the 9th Congress of Brain, Emotion and Behavior in Sao Paulo, and Dr. Stephen Suomi, his PhD and Chief of Laboratory of Comparative Ethology of Science of Eunice Kennedy Scorvin, of the National Institute of Child Health and Human Development and of the National Institute of Health. Is that right? Yes. Okay. Dr. Stephen, we would like to know nowadays, right now, what's really giving you hope, emotion, and making you really excited about in your work?
Well, there are two topics that are keeping us very busy and very excited. The first one has to do with some basic aspects of development. For years, we've watched our rhesus monkeys grow up under different conditions, and we've found that how they are treated, what kind of caretakers they have in their first 6 or 7 months of life is very important, such that depending on how they are reared, they show differences in attachment, differences in emotion, fear and aggression, and not only do you see effects of their early caregiving experiences in terms of behavior and emotional regulation, but you see it in terms of how their hormones work, especially their stress hormones, how their neurotransmitters are metabolized. And when you do neuroimaging of their brains, you find that their brains act differently functionally and are structured differently, have different wiring depending on whether they grew up with their biological mother or with some kind of substitute. And most recently, we found that these differences that occur as a function of early rearing extend all the way down to the genes, so that a large proportion of genes, over 4,400 of the 20,000 plus genes that rhesus monkeys have, like us, many of those 95% of the genes are exactly the same as ours. We now know that fully 1/5 of those genes are affected, both in terms of how they are expressed, whether they're turned on or off, as a function of how that infant was reared during its initial 6 months of life. And we now know what some of these genes do. We know what pathways are involved. And we now think that at least some of those genes are reversible, so that when you change the social environment of a monkey that's been reared under less than optimal circumstances, and for whom there are big differences not only in behavior and emotions and in hormones and in neurotransfers and in brain structure and function, but also in their genetic expression patterns, we can make those patterns more normal by certain types of social interventions. So that is one thing that's keeping us very busy and that we're very excited about, because if you can change genes in monkeys who experienced adverse situations when they were growing up early in life, then maybe you can do the same for humans as well. So this gives us some hope and some promise and some insights in how early experiences that are less than optimal in humans can be reversed, so that those humans can grow up to be more normal behaviorally, biologically, and in terms of their patterns of gene expression. So that's one thing, and we've been doing this kind of work for about the last 5 years. One of the consequences of this work is we've paid more attention to what's going on between mothers and infants in those initial days and weeks of life that might be part of the differences that you see when these infant monkeys get a little bit older. And what we found is that, contrary to what people thought in the past, that baby monkeys and their mothers show extensive face-to-face interaction during their first month of life, a pattern of interaction that was previously thought to be uniquely human. But now, when we look for it carefully in our monkeys, we can see it in that first month. The difference between humans and monkeys is that in humans, that face-to-face interaction is maintained for weeks, months, and years, and is thought to be the basis for the emergence of language, the development of theory of mind, the acquisition of capabilities to be empathetic, to be engaged in self-awareness and self-reflection, things that monkeys don't do. Well, for the monkeys, that face-to-face interaction stops after a month, and then they start doing other things. So it may be that that initial period, that initial month, is enough to establish a relationship between a mother and infant. And then for the monkeys, other parts of their social environment become more important for them. Whereas for humans, of course, they're still highly dependent on their mothers and other caregivers to survive. So now we're looking at some monkeys who fail to imitate during that first month of life, and we're finding that they are at risk for developing problems in social communication later in life, and in particular, some of them start developing autistic-like behaviors if they fail to imitate during the first week of life, whereas none of the monkeys who have successfully imitated during the first week of life have ever developed any of these autistic-like behaviors. So we think we might have discovered a very early potential screen for risk for developing autism, at least in monkeys. And now I have colleagues in the human autistic research field who are starting parallel investigations in certain groups of high-risk human infants who are born into families where there are already older siblings who have been diagnosed with autism, and we'll see where this work goes. But one of the things we're now doing, which we're very excited about, are interventions during that initial week and month of life to see whether we can get non-imitators back imitating again, whether the brain patterns that you see under conditions of imitation that you don't see in monkeys who fail to imitate, whether if we can get those non-imitators to imitate, do we see the same normal patterns of brain activity, and when they grow up, do they show normal social activity in terms of their play behavior and their lack of autistic-like symptoms? We will have to wait for these monkeys to grow up, but we're watching with a great deal of excitement and interest. So these are the two areas that are keeping us pretty busy these days and are keeping us very excited about the work.
And in your idea from 10 years from now, what is going to be?
Well, 10 years is a long time, especially when you figure that what I just told you, we weren't even thinking about 5 years ago. So all of this new exciting work has happened in our lab in the last 5 years. If I could make a guess as to where I'd like to see things in 10 years, I'd like to be able to better link changes in gene expression with changes in how the brain works and changes in behavior and emotional regulation that are occurring as a function of having individuals both grow up, natural development, and also what happens when they go into different environments that we already know affects their behavior and affects the way their brains work. Does it also affect the way the genes work? And again, if we are successful in reversing the effects of adverse early experiences, how long are those reversals good for? Are those monkeys who we have presumably rehabilitated now set for a good developmental course for the rest of their life, or do we have to keep doing other sorts of interventions? And if you're going to intervene, are some periods of development, like puberty or later in life, more readily accessible for changing patterns of behavior, biology, and gene expression? So are there other windows of opportunity besides early in life when you can make fundamental changes in developmental trajectories? So working out those details is, of course, of interest, and better understanding what's going on in the mind and the brain of these monkeys when they're imitating and when they're not, and to what extent does that change when you intervene very early. Do you then get normal subsequent social and emotional development and all the biological and genetic and gene expression concomitants? Well, it's going to take us a while to find that out, and also my guess is in 10 years we'll have a few other things to start looking at as well. It's an exciting time to be studying development, both in monkeys and in humans.
Thank you very much for your contribution as a whole in science.
It's my pleasure. There's nothing I'd rather be doing these days.
Thank you.